Climate change and increasing population are expected to cause considerable impact to the Pacific Island countries' food security over the next century. Rising sea level at a rate of 10 mm a year, saltwater intrusion, and other related climate change disasters are threatening root crops such as sweet potato, taro, cassava, yam, where about 70% of the Pacific Island population had depended on. The estimated Pacific Island population was about 12 million in mid-year 2020 and predicted to increase further in the next century, which poses more pressure on demand for those food crops. To mitigate climate change and increasing population, transgenic research in root crops was carried to ensure food security for the Pacific Island country, current and future generation. In the transgenic research in root crops, it involves transgenic strategies to improve productivity, resistance to pests and disease, and tolerance to climatic adaptive factors to mitigate food insecurity caused by climate change and increasing population in the Pacific. Agrobacterium tumefacium mediated transformation is one of the strategies to create transgenic root crops. This strategy uses a soil-inhibiting bacteria called Agrobacterium tumefacium that infect plant cells and cause tumor, which is conferred by a 200 kilobase tumor-inducing plasmid known as T plasmid that contains inside the bacteria. This T plasmid gives the agrobacterium the ability to transfer its DNA into the plant's genome. Thus, using this natural ability of Agrobacterium tumefacium, scientists can introduce foreign genes of interest into the plant's genome. Basically, there are several steps involved in using Ti plasmid in Agrobacterium tumefacium as a transformation vector. At first, the T region of the T plasmid is cut with a restriction enzyme, then replaced with a gene of interest next to the appropriate promoter, along with another selectable marker called antibiotic promoter. Next, the T plasmid transfer from the cell into the plant genome involves leaf disc of root crops sent off signals that detect by the agrobacter, causing induction of virulence. Genes that make DNA to exercise out and with the help of another virulence gene enters the plant cells and randomly incorporate into the plant's chromosome DNA. This plant hormone incorporated with new DNA and grow into transgenic root crops inside a medium containing both plant hormones and antibiotic selection. After are transferred to grow in soil having all the new traits: high yield, resistant to pest and disease, and tolerance to abiotic stress, that is, high salinity. This transgenic research in root crops can help coastal communities in the Pacific to develop root crops that tolerate high salinity with high yield that cater for increasing population and also mitigate destruction caused by disease and pests. However, there are few disadvantages to Agrobacterium tumefacium strategy, which are complex and time-consuming, dependent to plant genotype, requirement of sterile condition for regeneration, and optimal cell density of Agrobacterium is required. Although climate change and increasing population threatens our Pacific Island food security, transgenic root crops can be our hope.